Okay, so introduction to veterinary general pathology. So pathology is derived from two Greek words. First is patho and second is logi or logos. So patho means suffering or disease and logi means study. So simply we can define pathology as the study of disease, but we can also define pathology as the study of structure and function of the body in disease condition. Okay, that is called pathology. So study of structure and function of the body in normal condition, that is called anatomy and physiology respectively, but in disease condition that is called pathology. There are 5 aspects of a disease. So a disease revolves around 5 aspects, that is etiology, pathogenesis, structure and functional changes, clinical significance, and result or termination. So I will discuss each of these. Okay. So first is etiology. Etiology, as we can already know, this meaning is the cause of the disease. Definition is the cause of disease is called etiology, etiological factor. Etiology is of two types: genetic or intrinsic, acquired or extrinsic. Extrinsic includes infectious, nutritional, physical, chemical, all of these. Okay, so we'll discuss these in later at the end of this chapter. So pathogenesis, progressive development of a disease from the time of its initiation to the time of its recovery or invalidism or death. Okay, so pathogenesis simply means the progressive development of a disease. How a disease is progressing once it is initiated till the time of its termination, and termination might take place in 3 steps. Only 2 are written here, but there are 3 types in which a termination of a disease can take place, that is recovery, death or invalidism. Okay, next is structure and function changes. So these are the morphological alterations. Structure function changes can also be called lesions. So these are the morphological and associated functional alteration in the tissues. Okay, so lesion, what is a lesion? Macroscopic or microscopic alteration occurring in the tissue as a result of injury. Simple. So any macroscopic or microscopic lesion alteration occurring in the tissue as a result of injury is called lesion. Okay, there are 3 types of lesion: pathognomonic, macroscopic, microscopic. Here we will discuss pathognomonic lesion. Okay, so pathognomonic lesion, it is the alteration which indicates without any doubt the cause of disease. Okay, so an alteration which indicates without any doubt the cause of disease is called pathognomonic. Remember what is the meaning of this statement? It means that we can simply look at an alteration or lesion and we can say that okay, this disease, this is the lesion of this disease for sure. That is the meaning of pathognomonic lesion. So these are the important examples which was given in the book. Okay, so first is button shaped ulcers in large intestine and swine fever. So we have to learn these. In there, button shaped ulcers are found in the large intestine in swine fever, zebra marking of large intestine in rinderpest, punch out ulcers in filariasis of abomasum, and negri bodies in the cells of hippocampus and cerebellum in rabies. So you have to learn these 4 examples. These 4 examples are extremely important. There are 2 terms which are used by interchanging them: symptoms and sign. So symptom, any feature of a disease which is explained by the patient to the doctor in layman term, and sign, features of a particular disease explained by a doctor to the patient or owner of that, okay, in scientific terms. So that is called sign. And symptom, which is explained by the patient to the doctor in layman terms, that is called symptom. Pathogenicity, it is the ability of an organism to cause disease. So there is a similar term which is called virulence, which we will be studying in microbiology, by the way. Okay, so severity of or degree of severity of pathogenicity, that is called virulence. Okay, now clinical significance. So this definition we will study later. So diagnosis, so art of identifying the disease by using various laboratory tests and by the signs and symptoms is called diagnosis. Simple. How we are able to identify a disease using laboratory tests and by using various signs and symptoms of the disease, that process is called diagnosis. Okay, that technique is called diagnosis. And next term is prognosis. So prognosis is a probable outcome of a disease, meaning it simply means that how we sometimes doctor might say that the prognosis of a disease is looking good, prognosis of that disease is looking not that good. So prognosis means how will a disease terminate, what will be the probable outcome of a disease, that is called prognosis. And last aspect of a disease is result or termination. So it can occur in 3 ways: recovery, death or invalidism. As already stated here, recovery takes place when the damage produced by a disease is repaired. That takes place when the defenses are overcome, body defenses are overcome, and the tissue damage is great. And invalidism, what is the meaning of invalidism? Invalidism is simply, these 2 terms are self-explanatory, but invalidism means that when the animal or individual is in a mid state that it is not able to recover completely or not able to die. So that is called invalidism. Okay. Now lesion, microscopic which can be observed only with the help of microscope, and macroscopic which can observe without the help of microscope. That is the naked eye. Okay. This I discuss. Postmortem. So how will you define postmortem? So postmortem simply can be defined as the scientific examination of the body of any dead animal or individual or bird to identify the cause of disease. That is called postmortem. But in case of animal and bird, it is called necropsy. In case of human, it is called autopsy. Mortality rate, percentage of animal died by a particular disease in a population. So what is important? Percentage, not number. Percentage of animal died in a particular disease in a population. Okay. Next is morbidity rate. So percentage of animal affected in a population by a particular disease. Okay. Next is biopsy. So removal of a small piece of tissue from a living body for examination. So what is the difference between autopsy or necropsy or biopsy? So necropsy means the examination of a dead body of animal, and biopsy means removal of a small piece of tissue from the living body of an animal for examination. It is done in case of cats and dogs. Okay. Next is morbid changes. So changes is occur in the body after that. That are called morbid changes. Simple, we have to learn these definitions. Next is branches of pathology. There are 12 branches. General pathology, which is concerned with the basic alteration, which we are currently studying, which is concerned with the basic alteration of tissues in response to injurious stimuli. Special pathology, application of general pathology to a specific disease. Systemic pathology, study of alteration in a particular system. Chemical pathology, study of alteration of body fluids due to a disease. Microscopic, comparative, nutritional, clinical, forensic, immunology, experimental pathology. So these are 12 branches of pathology. Next is syndrome, a group of symptoms which occur together, that is called a syndrome. Two sides of a coin. The terms which are used as two sides of a coin, that is health and disease. So health simply means a condition in which an individual is in complete harmony with the environment, and disease a complete condition in which individuals suffer from some discomfort. That is called disease. Okay. So now we'll discuss etiology in detail, that is the intrinsic factor. For example, species. In the book the term genus was used, but the correct term which should have been species. For example, swine fever is a disease of pigs only. Canine distemper is a disease of dogs only. So this is the meaning of this genus or species. Breed, dairy breeds are more susceptible to diseases than beef cattle. And dogs, brain tumor are commonly found in bulldogs and boxers. So these are the breeds of dogs in which brain tumor are most commonly found. So these are the example of breed, how the breeds affects the cause of disease or etiology of the disease. Okay. Now strain. So certain strains of animal possess resistance to certain diseases. For example, strains are created by breeders actually. These are not naturally present. So strains of chickens, for example, white leghorn line 6, these are very resistant to leukosis. Okay. Now certain calves are resistant to hydrocephalus, hernias in pigs, all of these. Age. So tumor are observed in older animals. Triangle cellular disease of young horses and cecal coccidiosis is a disease of young chickens. Basic examples. Now gender, reproductive disorder are more common in female than in males, obviously, because females have a more complex reproductive system. Nephritis in dogs, males are more commonly associated with nephritis, and in cattle females are more commonly associated with nephritis. Color. This is an important point because the pigment, the melanin in the skin tends to protect the body from ultraviolet rays of the sun. So for example, melanosarcomas, this type of cancer which is found in the melanocytes and it is of the skin, it is grey, very commonly observed in grey and white horses, but very rare in brown and black horses because it is protected by, the skin is protected by the melanin pigment. And all of this, idiosyncrasy, this term is important. So it is an unusual reaction to some substances. For example, when we are administering a drug to a laboratory animal and the body of the laboratory animal is showing some kind of unusual reaction to that drug. So that is called idiosyncrasy.